Hi, thanks for clicking on my talk. I'm really excited to tell you all about Angel1 and how it might be involved in human ribosome-associated quality control. So most RNAs in the cell terminate in this 3' hydroxyl that we all know and love. However, certain metal-independent endonucleases in random self-cleavage use the RNA's own 2' hydroxyl to break the phosphate bond rather than coordinate a water molecule to break it. This leaves behind a circularized phosphate species known as a 2' 3' cyclic phosphate. RNA terminating in a cyclic phosphate are poorly understood, in part because this end isn't ligatable with typically used RNA ligases and is lost during most library prep and sequencing workflows. So we know very little about the role of these species and the importance of their resolution. In a recent paper from the Martinez lab, the first human cyclic phosphatases were identified, one of which is the protein Angel1, which is capable of resolving the cyclic phosphate back to 3' hydroxyl. Because of how poorly understood this modification is, we were interested in what pathways Angel1 might function in. And so we generated a HEC293 line expressing flag-tagged Angel1 and performed IP mass spec to identify binding partners. We looked at enrichment over a control line, and we did this with RNase and without to distinguish between RNA and protein-mediated interactions. Angel1 pulled down with EIF4E, a previously described interaction. But interestingly, Angel1 pulled down with components of the ribosome-associated quality control system, which is responsible for resolving stalls during translation. We pulled down on with RAC1, which is important for stalling, LTN1, which ubiquitinates the nascent chain, and ubiquitin itself. We also pulled down with all 3 known components of the SKI complex, which is important for decay of the mRNA during RQC. As a control, we also pulled down on TOE1, a factor important for snRNAs, and found a known interaction with SNRP-A1 that wasn't present for Angel1. So what is RQC? mRNAs can be made incorrectly or become damaged. And when these problematic mRNAs are translated, they can cause the ribosome to become stalled, which locks up valuable cellular machinery in an unproductive state. We classically think of 2 categories of stall-inducing mRNAs. There's non-stop decay, which are those without stop codons, often created by improper polyadenylation events or endonucleotide cleavage, and no-go decay, where the ribosome is stuck on some kind of UV or oxidative damage, difficult secondary structure, or problematic amino acid sequences, which prevents it from reaching the stop codon. In either case, what's functionally sensed by the cell is a trailing ribosome catching up and colliding with that stalled ribosome. The collided ribosome presents a unique surface that's recognized by surveillance machinery and triggers a cascade of events that leads to ubiquitination and degradation of the nascent chain, ribosome rescue, and decay of the mRNA, which often begins with an endonucleolytic cleavage. This cleavage is performed by an endonuclease recently identified in yeast and C. elegans by the Green and Air Bear labs, respectively. The 5' fragment is decayed by the exosome in a process dependent on the SKI complex, and the 3' fragment is decayed by XRN1. Interestingly, recent work has found that this cleavage reaction is metal independent and has shown that the 3' fragment after cleavage has a 5' hydroxyl, suggesting that this cleavage reaction proceeds through a 2' 3' cyclic phosphate intermediate. Because of Angel1's reported activity as a cyclic phosphatase and our mass spec data, we became very interested in whether or not Angel1 had a role in RQC. And so we first wanted to ask what RNAs Angel1 associates with, which we did with eClip. We did 2 replicates of eClip and sequenced associated RNAs and then categorized them, looking at enrichment of the IP over input. We found an enrichment of coding regions and 5' UTR and a depletion of introns, consistent with the factor that monitors translation and regions of mRNAs engage with ribosomal subunits. We were next interested in examining effects of perturbations of Angel1 on RQC. And for that, we needed a system to examine the rate of decay of substrates of the system. We used a northern-based pulse chase assay to follow decay of a beta-globin reporter in a HeLa TET-off cell line. We took the wild-type beta-globin reporter and removed all in-frame stop codons to create a non-stop decay substrate that we called BGNSD, which should be a substrate of RQC. In pulse chase decay assays, beta-globin wild type is typically very stable with a half-life of over 600 minutes. However, without any stop codons, this BG-NSD reporter is highly destabilized. We have two lines of evidence that this substrate is targeted by RQC. First, we can reintroduce a stop codon a few nucleotides before the polyadenylation site. Unlike the wild-type beta globin and similar to the NSD reporter, this reporter has no 3' UTR. Addition of the stop codon stabilizes the reporter, which is consistent with the lack of a stop codon being the reason for destabilization. Second, we can use an siRNA-mediated knockdown to deplete SCIV2L, a component of the SCI complex. We find a modest but statistically significant stabilization of the substrate, which is also consistent with our reporter being a substrate of RQC. We next want to use a system to investigate the human endonuclease homolog N4BP2. And we found that depletion of N4BP2 stabilized this reporter as well, which is evidence that N4BP2 is the human ortholog. Our critical question, though, was what would happen to our reporter when we deplete Angel1? We found that depletion led to significant stabilization of the NSD reporter, consistent with having a role in this pathway. We observed the same effect with two independent siRNAs and with an NSD substrate based on another mRNA. To test if Angel1 is specific for RQC, we examined a reporter containing a premature termination codon targeted by NMD. While NMD is a distinct pathway, it is also dependent on translation. And we see no difference when we deplete Angel1, which is consistent with Angel1 being specific to the RQC pathway, and that stabilization isn't just an effect of translational repression or general RNA turnover. One enticing hypothesis for Angel1's mechanism is that a cyclic phosphate generated at this step would require resolution before a process of decay by the exosome. To examine this, we can make a point mutation in Angel1 that removes its activity as a cyclic phosphatase. The corresponding mutation was shown to directly abolish cyclic phosphatase activity in a related protein, Angel2, by the Martinez lab. We can then examine stability after rescuing with the wild-type version or the mutant. By overexpressing an siRNA-resistant Angel1 wild-type after knockdown, we see a partial rescue. However, with the cyclic phosphatase activity abolished in the EA mutant, we see no rescue, which is consistent with Angel1's activity as a cyclic phosphatase being necessary for this pathway. Although we can't rule out a distinct Angel1 biochemical activity as a contribution of Angel1 to this pathway, or the cyclic phosphatase activity at another step being responsible, activity here would be the most parsimonious explanation for Angel1's role in this pathway. And I think this is a really intriguing hypothesis that I'm very excited to see further exploration on. Furthermore, the role of cyclic phosphates in the cell is an exciting and underexplored topic. And given that this modification is a frequent product of RNA cleavage and processing, I believe that this work may raise interesting questions about the widespread importance of removal of a cyclic phosphate to RNA degradation. And I'm really excited to see more work in this area examining that. So with that, I want to thank my lab, our collaborators, and funding. I would also encourage everybody to check out the posters by my lab mates Tiantai and Cody at the upcoming poster sessions. And at the talk, you're also very welcome to come find me and we'll chat about other questions. Thanks.